Okay, hi, this is your video for C3. So C3 is going to be the interaction and interdependence of organisms. So it's essentially looking at kind of the body systems that oversee some of the rest of your body systems. So it's kind of the coordination of them, particularly C3.1. So C3.1 is integration of body systems. So we're going to do a little bit just up at the top, kind of clarifying what we mean by that, where body systems fit into our level of organization and so forth. And then essentially we're going to divide the page straight down the middle and do nervous system on one side and endocrine on the other. So it's basically nervous and endocrine system is what this dot point is about. Okay, so this levels of organization is really important. So that should be familiar to you because it's one of the ways that the course is organized. So this isn't new information, but I just want to quickly go back over the levels and then look at what that means in terms of this concept of emergent properties. So if we recall, and again, your actual course goes one level before cells because it does look at molecules. So molecules are not always living. So if we actually are looking at like the levels of organization of life, then we tend to start at cells. So cells, of course, are made up of molecules, but then cells join together to make tissue. So tissue would be like muscle tissue, like a single section of kind of clustered cells that are working together. And then, of course, from tissue, we have organs. From organs, we have organ system. And then from organ system, we have organisms. So, of course, in your course, we've kind of skipped because the first level, we have molecules, then we have cells, and then this is now organisms. So we've kind of gone across here. And I would argue that most of this kind of topic 3 is really at the level of organ systems because most of what we're looking at is by individual systems. But that's kind of, and then we're generally talking about the organs within. But what we're looking at today is this idea that the organism needs to have some properties that are greater than the sum of its organ systems. So if I broke down the organ systems, I could say, okay, your circulatory system is going to transport and deliver things. Your respiratory system is going to gas exchange. So I could go into all of that, and then I could say, okay, an organism is the sum of all of that, but then that doesn't really take into account like consciousness and kind of experience and kind of that lived human particularly experience. It's harder to speak to this in terms of other organisms. So this idea that there is some things that as a whole, organisms tend to have this kind of carefully kind of constructed behaviors is greater than the sum of its systems. So that's called emergent properties. Not going to spend a huge time on it, but it is in the syllabus. And the best way I can say of kind of when you think about emergent properties is thinking about the sum of the systems. There is still something greater than that, which is the whole. So your kind of ability to quickly react and interact with the environment can't be accounted for by any system alone, nor necessarily by the interaction between them. So that's kind of the idea that there is some whole concept. And in particular, when we talk about kind of humans, we're looking at that as this idea of consciousness. But there is some degree of that even with animals. If you think about your pets, they certainly seem to have kind of a personality and an engagement that couldn't be explained solely by the interaction between the circulatory and respiratory system. So how are these emergent properties possible? So how do we exist as a whole being when we have all of these separate systems doing the same different things at once? And so that's where this kind of comes into C3.1, which is that we have the different systems, but we need some ability to integrate them to allow for you to be functioning not as a set of systems, but as a whole person. So that's going to rely on integration between the body systems. So some degree of kind of integration, bringing them together is going to be crucial. And that is done primarily by these two systems that we're going to look at today. So this integration that's really important for you to have this organism level, for you to be a whole person, a whole entity, and not just some of your parts. You really need these two systems. So I'm going to set up the table, and then we'll just kind of periodically erase parts of it because I'm going to divide it quite a bit. I said that. I'm actually going to erase all of this just to give myself a little bit more room. So the done by should come below this. So like I said, the rest of C3.1 can pretty much be divided into either the nervous system or the endocrine system. Okay, nervous system, and again, use about halfway of your page when dividing it. I don't think I've used my board quite that well, but anyway. A little bit closer. And the endocrine system. So before we go into nitty-gritty, so once we go through what each system does, then for the nervous system, I'm actually going to divide it by parts of the nervous system, whereas for the endocrine system, I'm just going to divide it by hormones. So we have kind of a system of organizing it. But first, let's be really clear on how it actually integrates. So if these are in charge of integration, what does that actually look like? So for the nervous system, it's all about electrical signaling. So it's this fast kind of action potential messaging between kind of the brain and the body. So it's this kind of messaging between the two systems. So the way it's going to kind of react is that way. The other thing I want to distinguish is the idea that essentially your nervous system is going to be in charge of interaction with your environment, whereas your endocrine system is more in charge of homeostasis. Not perfect, like there are exceptions because when we look at adrenaline and epinephrine, it is responsible to some degree in terms of interaction as well. So there's a bit of overlap, but electrical signaling is going to be in charge of kind of interaction with the environment. And also kind of to some degree with homeostasis. So when we look at kind of the brain stem, it's going to be more in charge of that. So it's not a clear cut. They both together kind of allow us to both interact and remain in homeostasis. But particularly when we talk about that very fast electrical signaling, that's very much about interaction with the environment. These are going to be fast messages. They're local, so they only activate the neuron near it, and they're short in duration. When we talk about the endocrine system, this is now chemical messaging instead of electrical. Those chemical messengers are going to be hormones, of course. Generally speaking, whereas this is really about this speedy interaction, this is more about remaining kind of balance. So this is going to be more about kind of homeostasis, growth, kind of things like that. So these are going to be more of the slower processes. So these are generally slower, more widespread. So one hormone can have a wider range of reactions, and they have a longer duration. So you could have to kind of compare the two. So those are some good ways of kind of comparing it in terms of what is their primary thing. I would say this one has a little bit of both in both systems. So probably not as strong of a comparison because it's not quite that divided, but they certainly have a little bit more of a specialty. Okay, so from here on out, I'm going to do nervous first because it doesn't really make sense to go back and forth now. So once we've kind of compared them, I want to go through the three different types of the nervous system you need to know about and wrap that up, and then we'll come back to endocrine. Okay, so the most meaningful way from here for the nervous system is to divide it into the peripheral nervous system, which is understanding nerves, the central nervous system, which is understanding the brain and the spinal cord, and then we'll have a little bit on the enteric nervous system, understanding that kind of control over the digestive process. I'm going to start with peripheral because I think it helps to have established what the nerves are before we get to spinal cord. So that's why I'm going to start in that place. So first one is going to be the peripheral nervous system. And so the peripheral nervous system is the nerves that run along your body. So this is all kind of nerves outside of actual cells in the brain or spinal cord. And you do need to know what makes up an actual nerve. So we're going to look at an individual nerve. Sorry, I probably don't need that. So this is going to be a single nerve, and nerves are going to have protective sheaths because we really do want to protect our nerves. Neurons are not very good at repair, so we want to do everything we can to protect them. Inside this is going to be kind of clusters of neurons. And within the same cluster, you can have sensory and motor neurons. So we do not have nerves for sensory neurons, MERS for motor neurons. They can be kind of coexisting in the same space. So if we have a look at what might they look like, so if we look at a sensory neuron, a lot of times they have kind of receptors at the end. They have the axon. They often have a soma that sits like this, and then some kind of dendrites like that. So that would be the general appearance. It does not say that you have to be able to draw them, nor is it overly emphasize their structural differences. So you have to generally know that neurons are long and have dendrites to receive messages, but not necessarily a lot of the difference between them. So sensory neurons are going to be all about sending sensory messages from either receptor cells or receptor buttons at the end of their own cells. So sometimes the receptors are separate cells. In the example of pain, they are just the ends of a single sensory neuron can detect it. So depends on the type, and they're going to send that to the central nervous system. So first to the spinal cord, then to the brain. In the same one, we also have motor neurons. Motor neurons have the more, probably what you have learned as the shape of a neuron, kind of going back to your 10. So they have the more typical large soma or cell body at the end of the axon with dendrites coming off of that. So a little bit different than the sensory neuron. And so there's going to be our motor neurons. And your motor neurons are going to be sending commands from the central nervous system to muscles. And so if you remember particularly HL, a motor neuron will either send a command to the nearest motor neuron near it, or it will send it directly to the muscle. So the last motor neuron is going to ultimately be in contact with a motor unit, which includes the muscle fibers that are responsible. The other thing they would like you to know while we're here on this is that within each type, the neurons can be myelinated or unmyelinated. So just a reminder there. So this would be an unmyelinated one. If it is unmyelinated, then that means every action potential has to go through essentially every little sodium channel. More on that will be in C2. But it essentially has to kind of open all along the way. So this is an unmyelinated neuron. And as a result, it's going to be a slower action potential. That's not necessarily always bad. So again, if we are looking at using the nervous system for slightly more homeostatic mechanisms, the neurons don't have to necessarily send the message immediately quickly. It can be kind of a little bit slower, even if you're looking at something like enteric nervous system and the passage of food, that's not kind of needing supersonic speed messaging. So using unmyelinated neurons can be a way of offering a bit more precision in messaging over speed. But then we can also have our myelinated ones. So if they are myelinated, that means they have myelin sheath on the axon that's made by Schwann cells. Does not look like the term Schwann cells is in your syllabus, so I wouldn't worry about it necessarily. So there's your myelin sheath. And then what this means is that the action potential bounces between them. So if you look at that example there, kind of comparing the number of arrows, we can see that it doesn't have to make as many kind of changeovers. And so this is going to be a myelinated neuron. And what it's actually going to do is it's going to send signals between the nodes of Ranvier, which is the gap. So it sends fast messages between the nodes of Ranvier. And so that's going to be really effective if we are looking at things like sensory motor neurons that are involved in pain responses, reaction times, things like that, speed is really important. It's going to be beneficial to have that really quick processing of sending that. Okay, so that's everything on the actual peripheral nervous system. It's just to recognize there are two different types of neurons, that they do look slightly different, but you don't necessarily need to worry about the details. What you do need to worry about is their functional difference. So sending information about sensory input versus sending commands. So that difference there. And then you do need to know that they belong together in a nerve and that they can be myelinated or unmyelinated. Okay, I'm going to erase everything under nervous. Okay, so I'm writing way off to the side. So keep yours in your spot, but I'm just going to move over a little bit so I can see myself on the screen at the same time. So you're still in your little table to the side, but I'm just giving myself a bit more space. So next up, we need to look at the central nervous system. This is probably the trickiest one to work out how much you really need to know about the brain, because this is a new course. Previously, it was a part of an option, and the option did have quite a bit of detail on the brain. It doesn't look like you have to know as much detail here, but I'm going to err on the side of slightly more detail. I don't think you're going to be asked what the lobes do, but I think understanding the idea of consciousness being the combination of these cerebral lobes is worthwhile. So I am going to kind of put it in there, but I would be surprised if you got point blank asked, but again, it wasn't the old option. So I don't know, I'm trying to do a little bit of predict work here. It doesn't label the lobes, but it does say you need to understand kind of conscious versus unconscious. So to me, that helps a little bit to understand kind of what's going on in the cerebral cortex. So up here, this whole kind of part up through about here is the cerebrum. And then within the cerebrum, the kind of technically there's a little space below that would have like the hypothalamus and things like that are actually not kind of part of consciousness. But most of the cerebrum is kind of the cerebral cortex or hemispheres. And that's going to be the four lobes. So it looks like in terms of what you would have to know, it is essentially that part. There's a few more things in terms of thalamus, hypothalamus that you don't need to know. So if we look at the cerebral cortex, you do want to know that that is basically in charge of consciousness. So consciousness is coming from the cerebral cortex. All right, so the cerebral cortex, a way of understanding consciousness is to look at the four lobes. So that's why I'm going to kind of go through them quickly, but just understanding what that kind of looks like. So there we're going to have the frontal lobe. And the frontal lobe is going to be movement and also decision making. So I'm just going to put movement and thinking. So if you are thinking about it, it is coming from your frontal lobe. So a relatively small part of the brain is what you know you were thinking about at any given time. From there, you have the parietal lobe. Your parietal lobe is going to be in charge of kind of touch sensation, touch, temperature, pain. So anything that is kind of not vision and hearing, but is related, or taste, so basically just touch, is all related to that. So it's essentially all things related to touch sensation. At the back of your brain, you have the occipital lobe, which is for vision. And then here we have the temporal lobe. And your temporal lobe is going to be for hearing. It's also going to be for memory. It's also going to be for emotion. So it's a really quite interesting one that, again, is part of that kind of emergent concept. So particularly if you look at kind of the frontal and temporal lobe, they can account for a lot of what is our ability to learn and kind of remember and have kind of conscious emotional experiences. So two really important ones there. And again, I would be surprised if you got point blank asked lobes, but you might get asked what the cerebrum does. And so knowing the lobes is a way of answering that in terms of kind of the sum of all of that is what it does. You could be asked point blank about this little guy here, which is our cerebellum. So it does have its own little dot point. So the cerebellum is going to be in charge of coordination and balance. So it allows for kind of your motor commands to come kind of separately per limb, and then it puts them all together. And then this remaining part here is going to be your brain stem. And you do need to know about your brain stem, but you only really need to know one part, which is the medulla. So the medulla is part of the brain stem, and that's the part that you need to know about. It has other parts to it, like reticular activating system and pons and all that. You can just think brain stem equals medulla, and that is in charge of the control of our vital functions. And you do have to know a little bit about the control over vital functions. So this is where the course kind of bounced around a bit, because then we went to endocrine, but then we came back to control of vital functions, but that's really a nervous thing. So I'm going to put it here with the nervous system because it's not hormone based. So let's kind of pause on this and say, okay, well, how does that happen? How does the control of vital functions occur? So the control of vital functions relies on receptors that actually detect kind of changes that need to be addressed. So you need to know about the two different types of receptors. So it uses two receptor types. So not two total, but two types. So it uses baroreceptors. So baroreceptors are going to be in charge of looking at arterial pressure changes. So these are going to be arterial pressure. So they are actually measuring the pressure on the wall of your artery. And that is going to enable your body to regulate your blood pressure. There's another word that it uses for that. So it is going to help you regulate your stroke volume, which basically means the amount of blood. So the way that it manages your blood pressure is by adjusting how much blood is actually going in and out. And if you regulate that, that reduces, kind of changes the pressure needed. So it regulates your blood pressure by regulating your stroke volume, by adjusting how much blood is actually going in at any given time. The other one is your chemoreceptors. And your chemoreceptors are looking at, again, the chemical composition. So that's going to be your pH, your CO2, and your O2 levels. And from that, based on that, it is going to regulate your heart rate, and your breathing rate, which is officially your ventilation rate. You can say respiratory rate, but I think they prefer ventilation to distinguish between kind of respiratory and cellular respiration and so forth. All right, so again, one little bit more on how that might look like is let's consider pH. So if we look at pH as our example, if you have an increase in activity, like exercise, the result is going to be an increase in your CO2 levels, because as you're exercising, you're using more cellular respiration. If you have an increase in your CO2, that's going to be a decrease in the pH. You should know that. That comes up in a lot of different topics. That increase in pH is what gets detected by a chemoreceptor. It could also detect the change in CO2. So remember, we do have other receptors to make sure we're not only watching pH, because it is sensitive to what your kidneys are doing at the same time, but we do have more pH sensors. So that's usually how it's done. When that is detected, that is going to send a message to the medulla, or it's going to be detected in the medulla. So chemoreceptors are both in your heart and in your medulla. And then that is going to kind of cause a stimulation of your diaphragm. And then if you increase the diaphragm contractions, that is going to increase your ventilation rate. So just kind of connecting that that's essentially how your brainstem works. So the medulla is going to actually be who actually does this. So the actual diaphragm contractions, that message is coming from the medulla. So the detection is either happening in the blood, but then sending a message to the medulla, or the actual detection is happening in the medulla as well. So it just kind of depends on where the receptor is that picks up the message. Okay, so that is all things brain. We need spinal cord as well, but I need a bit more space, so I'm gonna erase. And don't worry if you're running low on space, like I only needed this much, so you should be, you having used already most of your space is normal. So this is still central nervous system. Okay, so still on the central nervous system, we are going to add the spinal cord as another component of that. So here we have our spinal cord. And the neurons inside your spinal cord are going to be called interneurons. So a unique kind of neuron. And they tend to be very dendritic. So very kind of message oriented. So they have shorter axons and large amounts of kind of connective dendrites and terminals. So very, very branched. I didn't put what kind of neurons are in the brain because actually it's a bit complicated. It involves other types of neurons that you don't need to worry about right now. So just need to know about the interneurons primarily in the spinal cord. Your brain has interneurons, but also has sensory neurons for like occipital lobe and things like that. So it's a little bit more complicated. Okay, so what you need to know about in terms of the spinal cord is the pain reflex. So you need to be able to trace the steps that are involved in kind of managing that pain response. So first of all, it is detected by a nociceptor, which is a pain-receiving sensory neuron. So the nociceptor detects the pain. That single neuron, that is one sensory neuron, so the nociceptor is a single neuron, fires the action potential all the way to the spinal cord. Okay, so that action potential is going to go all the way to the spinal cord. So then the spinal cord receives the message. So it is going to intercept the message, and it is going to then send a command through a motor neuron. So then the motor neuron is going to get the command. The motor neuron sends the message to the effective muscle. So the effector is always a muscle. And in this case, they wanted you to be able to trace the specific example of like touching a match. So it would be worth noting that the effector muscle is the bicep. So the bicep contracts. So those would basically be your steps. You can break it up however you want. So if you want to say sensory, basically say fires accidental to spinal cord, then say, motor neuron is sent out. You can do it as 4 steps, 5 steps, just kind of depends as long as you keep those key things in there. And remember, it will still send it to the brain as well because your spinal cord itself is not able to process information. So it's just the reflex. So the reflex is these steps that happen with the spinal cord only. But if you've had some sort of injury, you do then need to process what happened, store a memory of it, access your memory to work out how you deal with those things, and all of that would involve needing your cerebral cortex, so it will also go there. Okay, so that is the spinal cord. Last little part of the nervous system is to look at the enteric nervous system. And your digestive system's done a bit dirty by the new course because it used to have quite a bit of digestion in the old option, the option that we actually did, but most of it's gotten removed. So this is like literally the only time we do anything on digestion, which is a bit of a shame because it's an interesting area. The key understanding with the enteric nervous system is that this is a set of neurons that are not connected to the central nervous system. So they actually do not need overseeing by the brainstem or anything. They have their own independent work. So if you think about that, the other example we've talked about that is a bit with the sinoatrial node and the idea that they can independently initiate action potentials without needing the input always from your brain. So similar idea. So this is a cluster of neurons that are able to control the involuntary muscle. So they're separate to the CNS, and they control the involuntary movement of food. So anything that is voluntary is still going through your central nervous system. I think that should be self-evident that you are making a conscious choice of when to eat and to chew and to swallow and all of those things. So the voluntary components of eating still belong in the central nervous system, but this is looking at the involuntary movements. Said involuntary movement occurs by peristalsis. So that's a word you do need to know. So these neurons are basically in charge of peristalsis, and you can define peristalsis again as waves or sequences of smooth muscle contraction. So smooth muscle is involuntary, but again, it's not single movements, it's over and over. I'm going to do a tiny little diagram of it. Itty bitty. If you have space left, if you don't have space left, it's probably not something you absolutely have to have a diagram of. So just kind of see how you go. Okay, so here we have our food. So this is our bolus of food, particularly in the esophagus. A lot of times by your small intestines, it's a bit more liquidy than that. And so in order to make this happen, it's going to be a contraction here. So contraction. And then on the other side of it, it needs to be a relaxing. So it has to contract one set of muscles while relaxing the ones ahead of it, and that pushes it forward, and then it would be here, and then now it would need to contract, relax. So these waves of contract, relax, contract, relax. Okay, so that's basically all on the enteric nervous system. Really is just to know what it is and know that little bit of information on peristalsis. Okay, so now I'm going to erase that, and now we're going to move on to endocrine. This is an SL heavy unit, so I am going to do the first 3 or SL, and then I've included all the plant hormone stuff under endocrine, even though we often don't necessarily refer to it as endocrine when it is plant, so I'm still going to put it there. I'm just going to also, won't have to close it. I just closed the blind to try to get less shadow. Sorry, it's been a bit shadowy before. All right, so endocrine system. So we already did our little bit of what it actually is. So now we're going to go through essentially just hormone by hormone. So I think that's the most logical way of addressing this, is just to do it. There's really only 2 hormones you definitely need to know. Interestingly, ADH is not named for SL. It's going to be a big one for HL in the next topic, but you do have to be able to account for the interaction between the hypothalamus and pituitary. So it's a good example, so that if you were to have to do that, to give that as your example. So I'm going to include it, but just a note that if you're SL and you're like, I'm dying, give me one less hormone, you can cut out ADH because it's not named. The 2 that are point blank named are going to be melatonin and epinephrine. Okay, so hormone number 1 is melatonin. So for each of these, you kind of need to know where it is secreted from and then what it does. So if you can kind of go through and think, I know who releases it, and then I know what it does, then you're in good shape. And then also kind of what triggers its release. So triggered by night or lack of light. So it's going to be triggered in response to not having light anymore. Who is it going to release by? Is going to be the pineal gland, which is in the brain, is kind of in that cluster of glands near the hypothalamus and pituitary. So it's at kind of the bottom of the cerebral cortex, so underneath the consciousness part. So triggered by light or no light, released by the pineal gland. What are some of its targets? So its targets are going to be kind of widespread. So it's going to do a number of different effects, which is true on kind of a lot of the hormones, except for ADH. So that's why it's a nice example, because it's very clear what its target cells are. But in this case, it's going to be things like dropping your body temperature. It's going to be increasing fat storage. So it's going to kind of remove some of the glucose from your bloodstream, because you shouldn't be needing to be as active and converting it to storage. So it's doing all of these things that are supposed to kind of get you ready for bed. And of course, the main one is going to be fatigue. So it is going to cause you to feel tired. So it's going to lower kind of alertness levels. The effect of this, so as a result of all of these things, you can say the effect or the purpose, however you want to put it, is going to be for you to have circadian rhythms. So the goal is for you to have a consistent sleep-wake cycle, which is why it's triggered by nighttime. So by having it be sensitive to nighttime, the result is that it can actually keep you kind of sleeping at night, which is better from an evolutionary standpoint because it's safer that way. Okay, so that's first hormone. Second hormone that you would have to know, we're going to kind of check that we know the same kind of things about this one. So the next hormone, I don't know why I boxed hormone instead of the name of the hormone, but is epinephrine, which is adrenaline, but we're going to call it epinephrine because that is their term of choice. What is it triggered by? So it is going to be triggered by any perceived danger, something that is seen to be a threat. So something that is received as a threat. It is released by, and let's check this carefully. So the release by is a little bit more complicated on this one. So the threat is actually going to be detected by the hypothalamus. So again, you could have to essentially describe the hypothalamus and pituitary. I would probably use ADH for that, but if SL, if you want to drop ADH and think you could do a good job with this one, that's fine. The hypothalamus then alerts the pituitary. It's actually the anterior pituitary. The anterior pituitary releases another hormone. That hormone, you don't have to know the name. It's like, I looked it up, I'm gonna forget it again. It's like adrenocortico something. Hormone, so ACTH. If you learn the name, you can call it ACTH, or you can just say releases a hormone into the bloodstream. So remember, this is the hormones always are gonna enter the blood. That hormone is gonna travel via the bloodstream to the adrenal glands. So that is technically its releasing agent. So the adrenal glands are who are going to release epinephrine, but you do need to account for the steps that get to that point. So danger is detected by the hypothalamus, alerts the anterior pituitary, releases another hormone called ACTH, which travels via the blood to the adrenal glands, and then the adrenal glands release the epinephrine. What are the targets? In general, the targets are going to be muscle. So it's going to be any number of muscles, but that is going to include things like cardiac muscle, diaphragm, things like that. There are other ones involved as well, but mainly it's going to be muscle related. So it's mainly going to trigger your cardiac muscle, diaphragmatic muscle to get kind of the increase in the oxygen levels. So the purpose of all of those things is going to be to increase rates of cellular respiration. In your skeletal muscles, so you want to have a situation where your muscles can get more ATP to be more active. And what is the purpose of that? The purpose of that is fight or flight. Okay, so hormone triggered by, released by targets. So I'm going to put yours underneath. I'm just going to do it right here. I'm just going to cheat so I don't have to erase that. So I'm going to do the last one, which is ADH. Like I said, this is a great example if you were asked to explain how the hypothalamus and pituitary can work together to regulate something. If you are HL, you definitely have to know it next unit, so it's a good one to use for this as well. If you're SL, you could try to use this instead, so up to you. Hormone ADH, which is antidiuretic hormone. I'm not overly concerned about you knowing the name at this stage. It is triggered by dehydration. So if you are dehydrated, you will be able to have that kind of trigger. It is released by, so the reason why I like this one for hypothalamus and pituitary is because you don't have another gland involved. So it's just hypothalamus and pituitary, so it's a little bit more straightforward. So the dehydration is detected by osmoreceptors. And those osmoreceptors are in the hypothalamus. And you can even go that little bit more and learn that essentially they change size depending on their absorption of water, and that causes them to send or not send messages. So when they're dehydrated, they shrivel in size, and that tends to cause signaling. Whereas if they swell, they stop signaling. So that way when we are hydrated, the osmoreceptors swell up and stop alerting it. So interesting things there. So the osmoreceptors are alerting it, but then they are going to then send the command to the posterior pituitary. So you have to know this pathway, but the pituitary doesn't play the primary role, whereas for ADH, the pituitary is the releasing gland. What is the target? This is also a nice one because its target is straightforward. Is going to be the kidneys. Forget other people have two. So kidneys are going to be the targets, and then the effect is going to be that they are going to reabsorb water. So they're going to take the water out of your urine and put it back in your blood. And the purpose of this is to maintain isotonic blood. So you want to maintain this kind of regular osmolarity. So maintaining an isotonic blood environment or monitoring osmolarity, which is kind of a more general term for the homeostasis regarding water levels. Okay, so that is it for SL. For HL, we need to do 3 plant hormones as well. So we'll keep going. SLs, you can pause or fast forward a little bit. You will need all of the immune system, which is next. All right. Okie doke. So now we are up to plant ones. So I'm just going to use the term, instead of just writing hormone like I have been doing, I'm going to use the term phytohormone. Because remember, a phytohormone is a hormone that is in plants. So that's what that means. There's only one that you need to know in detail, and you do need to know it in detail. So we will start with auxin and go into detail. The others, like, I literally just have a tiny bit of room, so you can use most of the room, just a little bit on cytokinin and ethylene at the end, but we'll focus on this one. So I'll start with the general information about it, and then we'll go into how it actually works. So what is our general information is that it is released in shoot tips. It travels in phloem, and it causes cell elongation. So those are kind of your 3 facts that I would know. So where is it made? How is it transported? What is its effect? One of the things it can do with the elongation is that it can cause elongation towards a stimulus. So by doing that, it can actually cause a tropic response. So in particular, you need to know that auxin, in this case, is responsible for positive phototropism. Okay, you do have to know a fair degree on how. So because in class we did it picture by picture, I'm actually going for a fairly just numbered steps because I was running out of room. So I kind of am going to do 2 main pictures, one that kind of shows what's happening overall, and then one that's showing kind of what's happening on a cellular level. So I'm doing it a little bit less pictorially, but go back to your notes for the longer one. So when we say what is positive phototropism, so remember, positive phototropism is basically the result of elongated cells on the shaded side. So this is going to be elongation of the shaded cells. Sorry, I should have actually drawn a little sun. So to make that more clear, so here was the sun. Sorry, I probably should have gone. So these are the cells that were away from the sun, have gotten taller. So elongation of the shaded cells. So let's look at how that happens, because you do need to be able to account for that. So that is what happens. Then we're looking at how does that happen. And again, I'm going to do this fairly briefly, just because We've gone through a lot, but go back through your notes if you need more kind of detail on it. So the first thing is that auxin is going to be released by phototropins that detect the sunlight. But it needs to move to the shaded side. So it is released and then it is transported to the shaded side by special transporters called auxin efflux carriers. Sorry, I want to make sure I don't use the wrong, yes, carriers. Auxin efflux carriers. Those are just going to be pumps that are made in the cell that's exposed. So they're going to be making these carriers in the ones that actually are activated by the light. So the cells that are activated by light build the pumps to move the auxin away from them. So the auxin is produced, but then it's transported away from the sunny side. So sunny cells make pumps, and then they move all of the auxin over to the shaded side. So it's going to be transported by these carriers, and that's going to create a situation where the shaded side has higher auxin. Once there, then the auxin in the shaded side stimulates the production of proton pumps in the cell membrane. So again, I'm going to draw kind of over to the side one single cell that's going to have to kind of cover all of this because of my sad running out of space. Okay, so I'm now going to build a proton pump in the membrane, like so. Those proton pumps are then going to pump protons into the cell wall. I'm going to use this other word that they did use. I put it in the notes. Apoplast is essentially the cell wall, but also kind of the space between cell walls of connected cells. So it's just the area outside the membrane that includes the cell wall and a bit of space between two cells. I think it's fine just to say cell wall, but apoplast, the syllabus does use that word, so you might want to just recognize it at least. So it's going to send the little hydrogens in here. Hydrogens are then acidic, and so that acidification of the wall activates expansion. So remember, there's a natural protein there called expansin that's already in the cell wall. And then expansin is going to break cellulose chains to allow it to stretch. So once the cellulose bonds are broken, it's now in a situation where it could stretch out. Remember, it does need turgor pressure for that to happen. So that's not going to happen unless there's turgor pressure as well. But I'm not going to write that down because I'm running out of space. And I think you're fine if you can just recall the rest of that. So here's all the little cellulose little crosslinks. And then expansion's going to come in. It's not going to break it all up totally, but it's going to break enough of the kind of crosslinks that is going to allow it to expand when water enters the vacuole. It'll push it out. So again, even if you had a 7-mark question, if you could say, what is positive phototropism? If you could kind of cover the idea that it's transported to the shaded side, makes the proton pumps, moves the H plus into the cell wall, activates expansion, breaks the cellulose bonds, cell is able to expand on the shaded side, which causes bending towards the light. So hopefully that should be enough that you could write easily on that without getting overly complicated on the gene expression part of it, which does look like it's kind of gone from this course. All right, I better make haste because I have to get out of here in 5 minutes, but we're going to try and do this between classes. Okay, last two things. I'm going to put them here, but you got to squish them below, sorry. Or put them on another page if you don't care about fitting it on a single page. So 2 more hormones you need to know about. So next phytohormone is cytokinin. Cytokinins. And again, I'm going to just do this part. So there's a little bit more. You could go into lateral growth, but I don't think you need that. So it is this time, when we look at it, it is released by root tips. It then travels in xylem. And it also is going to help with growth, but instead of doing elongation, it causes cell division or proliferation. So it's going to essentially trigger it to go into mitosis. That should be easy to remember because you should remember cytokinesis is part of cell division. So cytokinins trigger cytokinesis. So they're going to cause cell division. So root tips in xylem and cell proliferation. So ideally it has both. It has the ability of elongated cells, but big long cells are not going to be good for diffusion. So you also need cytokinins to start causing cell division as well. All right, and then the last phytohormone is ethylene. And ethylene, all you need to know is that it is released by ripening fruit. And then it also causes more ripening in a positive feedback loop. So it's an example of a positive feedback loop. That's most likely the context that you would be asked about it in. And that contributes to causing clustering of ripening. So a whole set of fruit that's near each other all ripens so that a herbivore comes and grabs one piece and takes the rest. All right, that's a lot I know on that page, sorry, and a lot of writing. So when we come back, we'll do the immune system. It's probably a little bit less writing, so a little bit more straightforward, and then we'll be done. Okay, so the second half of C3, which is still under this kind of interaction, interdependence, is the immune system. So the idea here is that for the rest of the body systems to function effectively, they need to be free of kind of infection due to pathogens. So if our body systems get compromised by infection, they can't function properly. So the immune system is kind of this protector, overseer of the other systems. So carefully, before we get way into the actual immune system itself, let's talk a little bit about pathogens. So when we say disease, we are specifically talking about the category of infectious disease. So disease can be used more broadly, but generally we tend to say disorder more for things that are non-pathogenic in cause, and disease for more things that are infectious in nature. Infectious just meaning we get it from another organism, so we're infected with something. And generally speaking, things that are infectious have a higher risk of also being contagious, but not necessarily. You can get infected with something that you're not able to pass to others. And infectious diseases are caused by pathogens. So we do need to know that term, pathogens. So a pathogen is anything that is capable of causing infectious disease. So fairly broad term that we're using there. And just a note of what all things can be pathogens. So a pathogen can be anything. So it can be viruses. So the main two that we think of are going to be virus and bacteria. But the other two that can also be associated with that are protists, amoebic dysentery, most common one is malaria, and then also fungi. So there's a few fairly usually benign fungal infections, but there are some instances of more serious fungal infections as well, usually more opportunistic infections. So any of those can be classified as that. The other word we need to be careful of is antigen. So pathogens have antigens. So pathogens are something that contains an antigen, but not just pathogens. So your own cells have antigens. So being careful with that, the antigen is not only to pathogens, it's just part of that. So antigens are cell surface markers. So pathogens have unique cell surface markers, and they're usually glycoproteins. So they're usually glycoproteins coming off of the cell membrane. And we call those specific to that pathogen glycoproteins antigens. So antigens are just whatever markers are unique to that organism. So if the pathogen was a little bacteria there, the antigen is the little cell surface markers. And I'm drawing a fairly generic triangle, which is not very helpful because it may make it seem like lots of things would have said generic triangle, but that would not be the case. It would be specific to that actual pathogen. Okay, while we're talking about pathogens, let this camera move over more. Let's look at what pathogens you need to know about. So you need to know a couple of pathogens by name, and the ones that you need to know are all in this category of zoonotic diseases. So one category of other general ones is zoonotic disease. And zoonotic diseases, think zoo meaning lots of animals. So by definition, a zoonotic disease is something that crosses species barriers. And we're basically looking generally at ones that infect humans, but that we get from animals. So transfer of pathogens between species. This is another one where the course is very specific. So at the end, I want to go through every time it names something super specific you have to know. So they have picked 4 zoonotic diseases that you need to know. I'm just going to revise the origin of it. It doesn't necessarily say you have to know the symptoms. So mainly we'll just focus on the origin for this component. So tuberculosis is transmitted to humans from cattle, often through their milk rather than direct exposure to them. Rabies should be one that you've heard of, and it is done through mammals, often domesticated mammals, dogs being the primary candidate, but can be from rats, other things as well. Japanese encephalitis. And that is the kind of interesting one because it is passed to us through mosquitoes, but the mosquitoes have to have gotten it from a bird or a pig. And we are dead end hosts, so that means we cannot give it to another mosquito or another person. So that's a distinct thing about that. And then, of course, COVID-19, which is believed to be initially from bats, but probably then through mammals before it got to humans, most likely. But the age when the textbook was written doesn't really have the whole story of it, so bats is fine. While we are on COVID-19, I'm just going to make a little note here so that when you're revising, that there is a dot point that says that you would need to be able to calculate percentage change versus percentage difference with data related to that. So remember, percentage change is between 2 time points. So between then and now, so what are the rates of COVID between 2021 versus 2025? Percentage difference is same time, often 2 sampling places. So that might be between transmission rates in UK versus America. So comparing two different things at the same time, you only want to vary one. So that's just a little random dot point. So I thought we would put that in there just so when you're revising, have a quick check that you remember that. Okay, so now what we're going to do is basically look at kind of the three lines of defense. So it calls it primary defense, but then it just says innate versus adaptive. So the course language doesn't really stick with 1, 2, 3. So I'm gonna kind of match their language and do primary, secondary, adaptive, which is not super consistent, but it's good to match how they're labeling it. So let's start with the primary defense. So your primary defense is to not let the pathogen in the first place. So the best defense is always offense. I don't know if that's the time, and maybe the best offense is defense. I don't know anything about basketball, but the idea is don't let it happen in the first place. So the best thing you can do is prevent rather than treat. So this is going to be our first level is primary defense. Okay, and so when we say primary defense, we are basically preventing, oh, I also can't spell defense. So not only do I not know the slogan, I can't spell it. I really feel like it should have an S. So what we are doing is particularly preventing it from access to living tissue. So that's a good way of thinking of it. Instead of saying access to the body, we really wanted to not have access to living cells. So we're preventing pathogen access to living cells. So pretty obvious that by far the main way we do that is through skin. So skin covers almost all of our body, and the epidermis, the outer layer of your skin, is not living cells. So your skin is covered in a layer of unliving, dead, previously living cells. You do have living tissue underneath that layer, but as long as the pathogen only gets on the top layer, you are safe, and there's no blood supply to the top layer, which is why it's not living. There are certain areas of your body that cannot be covered in skin. If you were to cover your mouth and nose in skin, you would not have any option for gas exchange. So you have to have some areas where there is movement of things back and forth. So in areas that do not have skin, they are covered in mucous membranes. And then mucous membranes often have cilia as well. And so the point of the cilia is to basically trap the pathogen in the mucus. And so if it gets trapped in the mucus, then it is not going to be able to get access to the body via the respiratory tract. There are some other ones as well. So the other possibility is like pH in your stomach, tears. So there's others as well, but those are just the two that are named. So following on from skin. So if we said skin is our primary one, then it's really, really important that we protect the skin and make sure that if it is damaged in some way, we limit access to the bloodstream and begin the process of healing that skin as quickly as possible. So the other thing that kind of fits into this is that we protect the body by clotting. So clotting does have a kind of a two-way point. So for one thing, it prevents blood loss. So that's kind of in some ways the most urgent, is we want to prevent there's not excessive blood loss. But in terms of this unit, the other reason for clots is to prevent access to the bloodstream from the external environment. So forming a clot of kind of dead red blood cells right away is a way that we can prevent access. So you do have to know about this in a bit of detail. So as always, the course loves to be a bit arbitrary as to what it wants you to know in excruciating detail. And this is one, not excruciating, but a fair amount of detail. So let's go through basically kind of 5 steps that are involved in this process. So the cut itself is going to activate platelets. And platelets are a good example of a positive reinforcement loop, whereby they tend to recruit more platelets, which recruit more platelets. So they activate lots of platelets. So they're going to activate the initial platelets, and that's going to lead to kind of an increase in the number of platelets, partly because of these clotting factors as well. So then the platelets that are gathered release clotting factors. And the clotting factors basically act as the starting point of a signaling pathway. So when we get to C2, we'll talk more about signaling pathways. This is an example of one, but you don't have to know all the steps in between. But the clotting factor is the initiator of a pathway of events. So this activates this, who activates this. So a series of events. So that causes, the clotting factors cause a signaling cascade, which again is just this knocks over this, think dominoes, knocks over this, knocks over this. The end point of the signaling cascade is going to ultimately be the conversion of prothrombin into thrombin. So you do need to know these terms. So prothrombin to thrombin. And the best way to think of this is thrombin is actually going to act kind of like an enzyme, an anabolic enzyme. So it is going to essentially add together fibrinogen. So here it is, this nice little enzyme. And prothrombin has essentially an extra kind of part to it that alters its shape and makes it not work. So it's basically like an enzyme with an inhibitor sitting on it. And so the end result of the signaling cascade will be the removal of the inhibitor. So thrombin is now active. So in this form, it would now be active. Once thrombin is active, it will then combine loose fibrinogen molecules into fibrin. So fibrinogen, I guess, would be the best way to say it. So then thrombin combines loose fibrinogen. It's not an oral exam, so pronunciation doesn't really matter. Into fibrin strands. So again, think of this little guy here. Here's thrombin doing the job. And then what it's actually doing is it's taking these that were all separate and joining them into a continuous strand. That's actually how the clot forms. So over here, here's my cut. What's going to happen then is these fibrin strands are going to come and form nets along the clotting area. And so fibrin here is what is actually going to create a net that traps the red blood cells. So it'll trap the red blood cells, and the red blood cells will get meshed into the fibrin and form a clot. So that is kind of what will eventually lead to the clot. Okay, so that's one area that you have to know a little bit of specifics about. So just check all your terminology, make sure that you are good on all of that. I'm going to go ahead and do secondary defense before I erase because it's tiny. So secondary defense is actually really interesting. So you could get all into the different types of phagocytes and look at neutrophils, and then kind of going to macrophages. You can look at sepsis, inflammatory response, fluid loss from vessels. So really interesting area. Sadly, you don't have to know very much about it. So I think I taught you beyond what you needed in class because I find sepsis really important. But you don't have to know that term. So now I'm going to condense it back to what you actually need to know of secondary defense. And you actually do not need to know the inflammatory response. itself. So the inflammatory response is important because it's essentially increased blood flow to the area. Increased blood flow to the area is going to be increased white blood cells, including phagocytes. So it is significant, but you just need to know about the actual phagocytosis part of it. So we'll go straight to that. So phagocytosis is going to be done by any sort of phagocytes, which are a fairly generic group of white blood cells. So it could be a neutrophil. You don't need to know that term. It could be a macrophage. Might be worth knowing that term. But any sort of phagocyte is a white blood cell. So then what happens is they can extend their cytoplasm. So this is called amoeboid movement, which is how amoebas move, which essentially kind of extends out the cytoplasm. The benefit of that is that it is going to allow for, I don't remember which color I drew my pathogen in before. Anyway, here's our pathogen now. It's going to allow for the endocytosis of the pathogen. So phagocytosis is just a type of endocytosis. So it's just a specialized form of that. This is a cell, so I'm going to give it a nucleus over here. Ooh, nucleus. The other organelle that is significant in this that you need to remember is the lysosomes. So over here, the lysosomes are going to be ready to merge with the actual bubble of pathogen. So because it comes in through endocytosis, it will come in as a little bubble, and then the lysosome bubble will join and spurt its enzymes into the pathogen. And then afterwards, it can just get rid of the waste. So after it's all done, it will then exocytosis out the waste afterwards. So it will break it down and get rid of the waste. At the same time that it gets rid of the waste, it will, if it's a macrophage, it can also present the antigen. So this is after the event. It will dispel the waste and present the antigen. If it's a macrophage, neutrophils do not present antigens. So not all of them do that, but it's going to be important for the next thing that we're going to talk about. So we can talk about adaptive immune response initiation, and that little antigen is going to be important there. So that's why I went ahead and included it. So that is basically what you need to know about all of that. The other thing I want to do is just terminology. So this is our second defense. The other term they want you to know for that is that it is an innate defense. And there's two things to kind of recognize if you see that term. So if you see that it is an innate defense, first of all, that means it is non-specific. So it occurs the same way for all pathogens. So it does not tailor it to the pathogen itself. The other thing is that it is not based on any sort of experience or exposure. So it is not impacted by previous experience. So it will happen the same way every time. So phagocytosis will come along and just try to grab any pathogens without identifying them first, and it will repeat that process regardless of whether that pathogen's been in your body before or not. So it's a non-differentiated process. So again, I kind of use the analogy that it would be like protecting a castle. Your first thing would be to have like a wall around it. That's like the skin. Someone jumps over the wall and you have a guard there, and you just tackles him right away. He doesn't stop and try to research his motives and work out what his criminal record is and contact his home country. If possible, the best thing as soon as the intruder gets in is to stop him in his tracks as quickly as possible. So if this is successful, then it can prevent the need for a further response. So a lot of times when we think of this, we would think of like if you've ever had like an infection of a sore or something on your skin, it won't cause you to have a fever and feel really terrible, but it will usually get red and swollen in the area. So that means you're kind of sending phagocytes to the area. You're trying to get it taken care of right there so it doesn't spread through your bloodstream and get other places. So that's kind of an area where you can see potentially this at work, where it doesn't require the full kind of otherwise extent. Okay, so that is kind of everything on primary and secondary. The detail is really going to be on the next line of defense, so the third line of defense, but we're just going to call it adaptive because they haven't used that term. They've used primary, secondary, but not tertiary, so that's fine. So essentially this is a kind of goal is to use less of these. So the goal is to succeed with primary, you don't need secondary. If that fails, the goal is to be successful with secondary. If that fails, then we need a targeted defense. So now they were not successful at jumping on top of the guy, or they jumped on one and someone else ran through. Now they're hiding somewhere in the castle, and then it might be a good idea to work out what's the motivation, who are they, what are they wanting, what are their weak areas, and how can we kind of come up with a scheduled synchronized attack to this. So that is the adaptive immune response. The adaptive immune response will take a couple days to get going. Generally, there has to be fairly widespread pathogen in the body to initiate it. As we get into it, you'll see it requires kind of a two-contact experience. So it's not easy to kind of happen easily. So, repeating words. But anyway, so the point of that is, again, by the time you're going through an adaptive immune response, most likely you're starting to already feel some symptoms if you're going through that the first time and having to initiate that. Okay, so what makes it adaptive? Two things. First of all, it is specific to that pathogen. So it is pathogen specific. And then the second thing is that it is impacted by exposure, or it's following exposure. And it changes depending on which exposure it is. So it doesn't look the same every time because the primary exposure and the secondary, third, fourth, fifth, whatever look different. Okay, so what I want to do for this is kind of do the initial first. So I want to go through basically the steps that are involved in a primary immune response. Then I want to come over here and look at kind of what is the difference then with a secondary immune response. So I didn't draw, I used probably more like almost 2/3 of this space, so you don't have to draw a line straight down the middle, but leave a little bit of room to the side for us to then pick up immunity and second immune responses. So we're going to start with the steps for the initial immune response. So this is going to be the primary immune response and the steps that need to go through. You do need to know this in terms of each step. So this is, it's not super detailed. You don't have to know, for instance, the name of the complexes like MHC and things like that. So it's not like detailed, but you do have to know kind of what all has to be activated. Okay, so essentially two things need to happen at the same time, or around the same time. They both have to have occurred before you can progress to step 3. And that is that two individual cells need to be activated by exposure to a pathogen. So I'm going to put the B cell first, but it could be that the helper T and then the B, like it doesn't have to be this exact order. But a specific B cell, so a specific B cell that is sensitive to that type of antigen. So there's a lot of different ways that glycoproteins can look, and we have B cells that are specific to specific types of glycoproteins. So this is a specific B cell encounters the pathogen. So that is going to activate that specific one. So it needs to be a pathogen that is consistent with that B cell. So that B cell, its specificity matches the antigen of that pathogen. Lots of words. Okay, so here is my B cell here. And here is its own little looking outer thing. And then it comes into contact with this, and it is compatible with that. So now this B cell is like, ooh, there is an antigen in the body that looks like my little antenna. So my antenna have encountered that. So my little markers that are looking out for antigens have seen an antigen that is compatible with the type of cell that I am. So that B cell is now activated. But our immune response requires a two-step activation. And that's important because we don't sadly get into it in the course anymore. We used to do all this stuff on allergies. It's so cool. But essentially, we don't want to over-immune respond to everything, because if we do over-respond to things, we get autoimmune diseases, allergies, anaphylactic reactions, things like that. So we want to make sure that we're actually only developing this kind of immune response to things that are actually targets that we should be attacking. So because it'll cause fevers and things that are unpleasant for us. So we have a two-strike policy. So if this happens and that's all that happens, the B cell, nothing else further progresses. So the other thing that needs to happen at the same time is a helper T cell is also activated. And again, this should be a specific helper T cell. Is also activated, and helper T cells are activated by phagocytes displaying the antigen. So a helper T cell is activated by an antigen presenting phagocyte, macrophage, sorry, it is a type of that. So again, we didn't go into the sequence, but if a pathogen gets in your body, you start with neutrophils. And if neutrophils alone can take care of it, it will never present an antigen. So what that means is if a random B cell comes along and sees it, but you never get to the stage of macrophages being recruited, you'll never start an adaptive immune response. So if a macrophage has been recruited, it means your neutrophils have failed. So there's all these things in place to make sure that we're not overreacting to pathogens. So around the same time, here's my helper T. I always draw it with a little halo because it's so helpful. And he's hanging out and he runs into a macrophage. And that macrophage was presenting the antigen. So this time he hasn't come into contact with the actual pathogen himself. He's come into contact with that actual, with that antigen. So now the helper T cell is activated. So it's basically been informed there is a pathogen in the body. It is requiring macrophage involvement, which means things are progressing and we need help. Now, once that has happened, now there is connection between these two things. So now the activated helper T cell we can call it double activates. Or signals the B cell. This, once that has happened, now the adaptive response is initiated. So that has to happen before. So initiation there. So now my active B and my active T, who again are both activated by the same thing, come into contact with one another, and the T cell signals further activation with something called cytokines, which you don't necessarily need to know that term because it's not named in there, but it basically is a chemical signal that is going to alert that one and double activates it. It is now ready to go. Okay, before we go next step, because I'm trying to catch the little dot points as well. So first of all, I want to make sure that you know this term that both B cells and T cells are also called lymphocytes. they're a little bit unique from other white blood cells. So they are a form of white blood cells, just like neutrophils and macrophages, but they are made in the bone marrow. T cells are matured in the thymus, and they're often stored in large amounts in our lymph nodes. So that's where the term comes from, is because they have an involvement with the lymphatic system as well, whereas the other white blood cells that are not involved in the adaptive immune response are less associated with the adaptive immune or with the lymphatic system. So anyway, that's where the name came from. You don't really need the origin, but you just need to know that you could see like a B lymphocyte instead of a B cell, and it means the same thing. I'm just using B cell because it requires less writing, but you could also call it a lymphocyte. The other little dot point, just to kind of be aware of, is they also have a little note about HIV. So just a reminder that HIV virus attacks types of helper T's. It doesn't necessarily attack all of them. A lot of times it will attack ones that are sensitive to similar receptors. So it will attack a category of them, not necessarily all of them. Sometimes this attack can be more widespread, but it will cause a loss of a whole group of helper T's. So that is going to cause essentially low levels of helper T's. As these start to die out, because of that, you end up with AIDS, which is acquired immune deficiency syndrome. And then the other term that they want you to know as well, which is connected to that, is once you have this low level of helper T cells, particularly helper T cells that are beneficial for fairly and what otherwise would be fairly innocuous and common things. So you have helper T cells in your body for lots of really common bacteria and viruses in the environment, even like certain fungi that just we're in a lot of contact with. You tend to have fairly good helper T cells established for these kind of recurrent threats. But people that have HIV, they often lose those kind of established helper Ts, and then they basically succumb to things that otherwise are pretty readily treated in our body without any problem. And so the term for that is opportunistic infections. So again, opportunistic infections are things that in a healthy person, our helper T cells would pretty quickly be responsive and eradicate. But in someone without a healthy immune system because of HIV, they start falling to these kind of otherwise preventable illnesses pretty easily. Okay, so now we are starting our response. So the next thing that needs to happen is, because again, this is a specific B cell, I now need a lot more of that specific B cell. So it's going to be rapid cell proliferation by mitosis. So the activated specific B cell now proliferates by mitosis. And again, mitosis is time consuming in the sense that it does take about 10 hours or so for one cell divide into two. So if we were trying to get kind of an army, it is going to be a bit of a lag, and that is going to take a day or two to really get that kind of going. So this proliferation stage is going to take a couple of days. Once it has proliferated, then that army, the clone of B cells, which is just a group of them, are now going to differentiate into two types. So they are now going to differentiate into plasma cells and memory cells. So plasma cells versus memory cells. Most of them are going to become plasma cells. So you have way more plasma cells than you have memory cells. The plasma cells are going to start releasing specific antibodies that go with that pathogen. I'm going to actually erase and redraw that up at the top just because I've run out of space. Just for awareness, we're up to like here, so you don't need a lot more space, but I have run out. And I was gonna make it too small to be able to see what I was doing. Okay, so after mitosis, now that we have a whole bunch of them, now they are gonna differentiate, and we're gonna have plasma cells, and we're going to have memory cells. And again, most of them are going to be plasma cells. So what's going to happen is the plasma cells are going to start releasing antibodies. And the antibodies are going to be specific to that antigen on that pathogen. So these are the antibodies that are specific antibodies. And this little part here is the variable region, which means it looks different on every antibody, and it has to be compatible to the antigen on the pathogen. So variable region is compatible or shape match to the pathogen's antigen. So the plasma cells are just going to start mass producing these. So they're going to be essentially just making them and exocytosing them like wild. The memory cell, meanwhile, is going to hold on to the memory of how to make one, but it is not going to actually start producing them. So it's not going to be an active current production of antibodies, but it's like I have all the stored information of how to make these. So it is going to stay long-term in the body, whereas the B cell is going to eventually, once it's been used and it's done making the antibodies, it's going to be done away with. It's going to be broken down for parts. Okay. One other little random thing, so then what do the antibodies do? So let's look again if we were to have a couple of pathogens and they have their little antigens on them, like so. So what can happen is the antibodies can kind of cluster them together. And so it can cause essentially a clustering of pathogen. And then when they cluster, they alert the macrophages to come and destroy them. So the antibodies cluster and destroy pathogens. One little random dot point is that it has on there that this same process is why you have to have compatible blood types. So this whole kind of cluster and destroy, this same process happens if incompatible blood is given. So essentially, if blood types are done by antigens, and so we have antibodies in our body to any blood that is not compatible with our own. So we have antibodies there waiting. So if we get given incompatible blood, those antibodies that are naturally in our body will attack the blood, and that is how you will get hemolytic disease and things like that, where it'll start destroying the blood cells instead of the pathogen. So that's just a little kind of connector note to recognize that this is actually the same thing, because we have natural antibodies, like we have anti-A antibodies in our body if we don't have type A. So that idea, it's a little bit more complicated than that. We're going to get to it again with genetics and really clarify how the allele determines the antibodies. So the part here is just to recognize that's how they have the effect. Okay, last little thing here is just a little bit of a note on medical treatment. So all of this is happening and all of that is causing a natural treatment. So then the question becomes, well, what would the medical treatment be that could help in this process? So if you were trying to fight this off during this process of building and waiting for mitosis, and it's taking a while, how could your doctor support you? So the answer would be that you could take antibiotics. So antibiotics are medications. And just being careful that antibiotic is not the same as antibody, so all the terminology. So antibiotic are medications, and they target prokaryotic structures and processes. So a lot of them actually target the structures. So certainly initial ones tended to target like the specific cell walls, like peptidoglycan. Some of the more advanced ones are now having to try to help with a wider range of pathogens. So they tend to do more like the ribosomes. So if they can, target certain ribosome types and so forth, and certain metabolic processes that are only found in them, then that can be really helpful. So the reason why that's good is because it will not affect our cells. So it will kill bacteria, but not our own cells. So they're safe, they only target the prokaryotic cells. The downside that you need to recognize is that viruses are not living cells, so they are ineffective for viruses. And the other one that again it has a dot point about is just resistance. So I'm just gonna write that. I'm not gonna go all the way into it again because we've covered it fairly thoroughly under natural selection, but natural selection is leading to the resistance, which is leading to medications like this being less effective. Okay, so now let's talk memory cells. You probably need to draw a bigger arrow. Mine is gonna just sit right here, but yours is probably needs to go all the way up. So now we're back up into this little space I left. Okay, so what we want to look at is what role do these have? So memory cells cause immunity. So that is a set term they want you to kind of know and use. So when we say immunity, we basically mean resistance to a pathogen. Due to memory cells. And it's going to be due to previous exposure. And when we say resistance, the main benefit of resistance is going to be that you're not symptoms, no spreading. So no symptoms, no spreading. And those are both important. The no symptoms is important for you so that you don't feel bad. The no spreading is important for the community because once that's the case, you're no longer contagious. Okay, let's draw the graph now. So you probably have a little bit more room than this because you moved yours up a little bit, but so I'm just gonna cheat and erase this. So you do need to be familiar with this graph. It is something you could have to draw or interpret. You need to make sure that you know that this is the concentration or essentially the number of antibodies. It's usually done by concentration. It's just how they calculate it. So one or the other, and then down here is going to be time. Okay, so then what we want to compare, what we've done here, this whole thing has been on the primary response. So we're looking at essentially the first time you get something. So here we'll mark kind of first exposure. And so what we are seeing is it will be kind of a bit of a lag, to be honest. Or there'll be a bit of time before you really see any antibodies being made because you have to have the macrophage, alert the helper T cell, etc. So you have this kind of really slow kind of start. Then you'll start making them. And then it will peak at somewhere around kind of 7 days if you have a typical kind of cold flu, something that has a relatively quick transition between exposure and symptoms. So that would be somewhere maybe around a week, and then you now have max antibodies. They're going to fight off the infection, and it's going to go down to having no antibodies anymore in your active bloodstream. Then after that, you're then exposed a second time. And again, we talked about the fact that this happens all the time because we often give something to someone that we're in close quarters with, so we get re-exposed a few days later when they're now sick with it. But this time there's not going to be a lag. Almost as soon as your exposure, within a couple of days, you're going to get a response. Still might take 2 days because the activated memory cell still has to trigger mitosis to make more cells to create more antibodies. So it knows how to make them, but it will need its helpers. But you don't have to have the whole kind of waiting on activation. So you skip all the way down and then you initiate proliferation, but you can start, the ones that are being made can start producing antibodies right away. So it's still faster, but there has to be a couple-day delay. But generally speaking, that delay is not enough for symptoms to develop or for the load to become enough for you to be symptomatic. That's going to also make way more, and they're also going to take longer to go away. So kind of multiple effects there. So what we want to note is this is going to be here. This is the primary immune response. So this whole experience here, primary immune response, and what we see here, this is the secondary immune response. And so kind of 3 differences that we want to make sure to note. The first is that it's faster. So we see here there is a faster production of antibodies. So here we have kind of this lag, and here it's a much faster, and that faster response is particularly responsible for this. So the idea that there's not the time for it to kind of spread in our body. The next thing I would like you to note is that there is a larger response. So compare this to this. So there is also more antibodies made. And then partly as a result of the fact that there are more, they tend to also remain longer. So it takes a bit longer for us to break them down. So the antibodies tend to stick around a little bit longer as well. So those are kind of the things that you would need to recognize there, the 3 differences. And again, why has that happened? It's all because of memory cells. So our memory cells prevent the need for the 3 steps prior to that, and you can go straight to making antibodies, and then they'll be proliferating and making at the same time, so you can get that kind of faster response. Okay, when we say they are memory cells, we do want to add our last little point, is that the memory cells can also be the result of vaccines. The benefit with the vaccine is that you get the exposure without the illness. So essentially the primary immune response occurs, but it responds to the vaccine, which is kind of symptomatic causing, so you don't ever have to be sick from it. The reason why it doesn't cause the actual symptom is because it is usually a weak, dead cell, or even more common now is kind of DNA from, or RNA from the pathogen only. And it often is not the whole part. It might be the DNA just for the antigen. So we put in the DNA for the antigen, and then our body responds to that actual piece of DNA as it creates the antigen. And so we basically get the antigen without the pathogen. So the idea is antigen, no pathogen. It's kind of a good way of thinking about it. And the benefit is the antigen triggers the immune response. but because there's no pathogen, you'll get no illness or symptoms. You can get side effects, which is essentially just due to the immune response. So sometimes because your body is undergoing immune response, you will actually get some degree of symptoms. And then last thing, I'm so sad that I've run out of space. I'm gonna write it over here, but you just write it lower. I just don't wanna erase when I'm so close. Okay, so with vaccines, we also just wanna add that if enough in the population get the vaccine, and I'm going to say have immunity. It doesn't have to be through the vaccine. It could be that many people got the infection. So to be honest, when COVID was going well, the number of people who had COVID contributed. So it was vaccination, but also prior exposure. Then that causes what we call herd immunity. Herd immunity is somewhere around 70 to 90%, depending on a lot of factors related to the contagiousness of the actual pathogen. And once that has happened, it will basically die out of the population because of a lack of hosts. So I'm going to write the disease goes away. As we're seeing from measles outbreaks, doesn't truly go away, it just stops causing disease. So the pathogen remains, the disease seems to go away. So as long as herd immunity is maintained, then you generally don't see disease. In some cases, it can be completely eradicated. In some cases, it stays around in very, very, very low levels because even people who are vaccinated might get it and have, again, this kind of fast exposure, but it's still around. So it kind of depends on the pathogen. Some of them have been eradicated. Some of them just seemingly kind of go away, but are still around in very, very mild levels. And remember, some things like bacterial things, a lot of them cannot actually ever reach herd immunity. So things like tetanus that survive in the environment. Okay, I think that is all things defense. So this topic only has the 2 parts, so just the integration defense, both a little bit long in terms of how much there is in each one, but just the 2 components. So good luck.